Hi everybody, it is your AP Biology teacher, Mr. Poser. Today we are doing a Unit 2 AP Biology recap. So this will be the next one in the series of recaps of the whole unit that I'm going to be doing. So this is Unit 2. I'm planning on getting through to Unit 8. And I'm going to try to get through all 11 topics in Unit 2 relatively quickly. I'm going to try not to ramble too much and just get to the point and move on. All right, so here we go. Let's start. We're going to start with obviously 2.1. Some of the main ideas here is that all life is made of at least one cell. So last unit was all about the molecules of life, how they work, biochemical reactions, that kind of stuff, hydrolysis, dehydration, synthesis. Now we're getting into the unit of a living thing, which is a cell. And all life, in order to be a living thing, you have to be made of at least one cell. And thus cells are the smallest unit of living things. You can categorize cells into two groups, whether they are prokaryotic or if they're eukaryotic. Prokaryotic cells, do not have a nucleus or do not have membrane-bound organelles. In other words, they are not compartmentalized. At the end of the video, we're going to talk about what that means. So like bacteria and another domain of organisms called archaea, some of the very first organisms to arrive on planet Earth. I shouldn't say arrive, evolve on planet Earth. That's why they're called archaea. Archaea means old. Are prokaryotes. Eukaryotes, like you and me, eukaryotes, have a nucleus and membrane-bound organelles, and these are protists in multicellular life, aka fungi, plants, and animals like ourselves. Our cells are eukaryotic, and they do have membrane-bound organelles and a nucleus. However, every single cell, doesn't matter what you are, has DNA, a plasma membrane, cytosol, and ribosomes. A plasma membrane is the outer border of a cell that controls what goes in and what comes out. We spend a lot of time on that in this unit. Cytosol is the fluid that fills in all the space in the cell. It's also known as cytoplasm, depending on who you ask. And then ribosomes. Ribosomes are responsible for using DNA to make proteins, and we spend a whole bunch of time on that in Unit 6. So that's what those three are. Here's our prokaryotic cell. DNA kind of just floats around in the cytosol rather than compartmentalized in a nucleus like in a eukaryote. So yeah, that's what these three things do, and every single cell has all three of those. But eukaryotes have those three structures and membrane-bound organelles. So all the organelles right here, all the structures of a cell are what we call organelles because they have their own membranes surrounding it, and they're made of at least one membrane. Right, so a nucleus has a double membrane called the nuclear envelope around it, while some of these structures are just systems of membranes, and we're going to go through each one very quickly here. The nucleus, as I said before, contains cellular DNA and it contains chromosomes. We're going to spend a lot of time in there in the next couple of units when we get into genetics and genomics and protein synthesis. Right, nuclear envelope is the nucleus membrane, and it's continuous with the smooth and rough ER, which we're going to look at here in a second. System of membranes surrounding the nucleus. And the nucleolus, the innermost part of the nucleus, produces ribosomes, and that's pretty much your anatomy of the nucleus. And again, that is only in eukaryotic cells. The ER, endoplasmic reticulum, is a network of membranes responsible for synthesizing products and packaging them in vesicles, aka storage or shipping units, kind of like little bubbles of membrane that a cell can put a protein in or a lipid in, whether it's a hormone or a signaling molecule or an enzyme, something like that. It can be packaged into a vesicle and shipped out. So the rough ER helps to make proteins because it's studded with ribosomes and ship them out in a little vesicle. And the smooth ER makes lipids and it ships those out in vesicles as well, and helps with detoxification of the cell too, to break down and convert toxic compounds into those that are more safe for the cell. That is what the ER does. The Golgi complex works in tandem with the ER. And once again, these are flattened membrane sacs that fold and quote-unquote package and ship cellular products like proteins and lipids from the endoplasmic reticulum. It is the shipping center of the cell. It says in the AP Bio curriculum not to use analogies to talk about the parts of the cell, but whatever. And there's not really, this is a good one, right? It's the shipping center for the cell. It's like the Amazon fulfillment center. Sponsor me, Amazon. Just kidding. The ER and the Golgi comprise what we call the endomembrane system. Endo means within, membrane means membrane, and system means more than one structure working together. So yeah, those three work together to make products and ship them out to the right places, whether that's in the cell or out to another cell, whatever. The mitochondria is the powerhouse of the cell. That's it. That's the video. Just kidding. Site of ATP synthesis and cellular respiration. Okay, I just had to put that in there. I'm sorry. It's just it's a meme at this point. Yeah, we're going to talk about more the mitochondria in just a second. All right, lysosomes, a membrane sac of hydrolytic enzymes that break down molecules. Hydrolytic, what does that mean? hydrolysis. Okay, it's a big watery bag of death that'll break down anything that comes into it. All right, so whether it is a foreign particle or a foreign cell or something, a piece of food that needs to be digested, and it's going to be sent to the lysosome to break it down through hydrolysis, okay, which is what we covered last unit. So hydrolysis occurs in the lysosome. These three structures here that we're going to talk about are only found in plant cells, only found in eukaryotic plant cells. And those are the central vacuole, the chloroplast, and the cell wall. All start with a C. The central vacuole is a large membrane vesicle that stores nutrients and water, and it controls the water and what we call turgor pressure in a cell. Hey, turgor pressure is important for plant cells. We're going to talk about this in a second when we talk about tonicity, but a plant needs to have enough water to exert force on its cell wall and stay rigid and stay what's called turgid, so a plant can stay up and it's not going to droop because it does not have enough water. The central vacuole plays a big role in that, and it's a storage unit as well. Okay, the cell wall, okay, I don't know why, but I have a lot of students that think that animal cells have a cell wall, and cell wall is really important and really, really strong, and like you need it to survive. And I'm like, yeah, you do, but like you're not a plant, so you don't have a cell wall. But a cell wall is a rigid barrier of outside plasma membrane that provides support and structure. Okay, so plants don't have bones. Okay, they're not able to stand up on their own without a cell wall. Okay, a cell wall allows them to, well, it helps them build the turgor pressure, and it also helps them stand upright. So that's what a cell wall does. And it's a protective barrier as well, kind of not super similar to a plasma membrane, but we're going to talk about that in a future topic here in a sec. Chloroplasts at the site of photosynthesis. They produce glucose using water, carbon dioxide, and light. Photosynthesis is a big 3.5 topic. I won't spend too much time here because we also talked about it in 2.2. So here we go. A big theme in biology is that the structure follows function. I just got done talking to one of my classes about this today, is that the way something is built allows it to do its job. And that's a huge theme in biology. If you take a class, human anatomy and physiology, anatomy is the study of structures, physiology is the study of functions of those structures. So the way something is built, the way something is structured, allows it to do its job or its function. And subcellular components are no exception. They are structured in a way that allows them to carry out their function. An example of which is a mitochondrion. The mitochondrion has two membranes. One is outer membrane that allows stuff in and out of the mitochondrion that's smooth, and then it has a super foldy inner membrane, highly folded, and those folds are called cristae. These folds, what they allow it to do is they provide surface area. We're going to talk about that in a sec, surface area for reactions to occur. But different phases of cellular respiration, which is what the mitochondria does, it makes ATP using glucose and oxygen, occur in different compartments of the mitochondria. So when we talk about cellular respiration in Unit 3, that's going to come back in a big way. So we have an outer membrane, inner membrane, and then the inside of the inner membrane is called the matrix of the mitochondrion. And something else about the mitochondrion that's notable, talk about that at 2.10, 2.11, is that it has its own DNA and ribosomes as well. Okay, so once again, the structure of a mitochondrion allows it to carry out its job very effectively, making ATP. Same with the chloroplasts. Chloroplasts also have two membranes. Those discs within the chloroplast, if you take a look at this picture here, there's a diagram. These discs are called thylakoids. And the thylakoids are the locations of what we call the light reactions of photosynthesis. And again, this is Unit 3. This is Unit 3 stuff where this is going to come back in a big way. And these stacks of discs are called thylakoids, but they are stacked called grana. A stack of them is called a granum. The rest of the chloroplast is filled with a fluid called stroma, which is the location of what we call the Calvin cycle. Different photosynthesis reactions occur in thylakoids versus the stroma. That's what I just said. And curiously enough, we're going to talk about this 2.11. The chloroplast also has its own DNA and ribosomes. All right, on to 2.3. All living things must be able to exchange material, aka nutrients and waste, with their environment. Okay, you eat food, you get rid of waste, you have to exchange materials with your environment. Same for every other living thing. It's one of the rules of being alive. So hence, if you're a cell, okay, you don't have specialized systems to allow us that allow us to take in food really fast and eliminate waste really effectively, and like, wow, I can breathe in oxygen and send it to my body super fast. Cells don't really have that. Cells are super small, and they rely on diffusion. And the reason why they're so small is because of diffusion. And this is going to come up again, but diffusion is the natural movement of particles from low to high concentration, which can be very, very slow. All right, things just kind of spread out naturally. If you put a drop of food coloring into a glass of water, hey, and just let it sit for a while, it's not going to spread out very fast. It will spread out eventually, but a lot of times you got to shake it up to like move it along and make it spread out faster. So diffusion happens pretty slow. But because the diffusion is slow and cells are small, a cell's surface area to volume ratio is extremely important. How much surface area it has on the outside of it relative to how large it is, how much space it takes up, aka its volume, is a very important concept here. So larger cells have a smaller surface area to volume ratio. Okay, they tend to have a larger volume. As volume goes up, surface area tends to go down. Okay, and a larger surface area to volume ratio is better for cells to exchange materials, take in nutrients, and eliminate waste. So cells can't grow very large because of the limitations of surface area and volume. Okay, but cells with higher surface area and volume are more efficient at exchanging materials. But a specialized structure called microvilli allow cells to increase their surface area. All right, once again, form follows function here, right? Say cells in the intestinal lining, the inside of the small intestine, the intestinal lumen, right? Your small intestine, you eat food, all right, your stomach breaks it down and your mouth breaks it down, but then that slosh is sent all the way through your small intestine, and what your small intestine does is absorbs those nutrients from what your food used to be, right? So it's going to be very advantageous for it to have little extensions here that are going to increase its surface area so we can interact more with its environment and absorb nutrients from it. Okay, so that's what microvilli are. Now, the AP Biology exam will ask you to know how to calculate surface area and volume ratio of a cube-shaped cell and a sphere-shaped cell. I'm going to give you the formulas. They're right here. They're also going to be on your exam. We're not going to do any practice problems right now because this is a recap and I'm trying to go fast. But the surface area of a cube is 6 times side length squared. Volume of a cube is side length cubed. Okay, you're going to have to compare those to calculate a surface area to volume ratio. So surface area divided by volume. Then of the sphere is 4 pi r squared surface area. And then volume of a sphere is 4/3 pi r cubed. Okay, and you're going to have to compare those once again to find surface area to volume ratio. Again, not doing any practice problems right now because we got to keep moving. On to 2.4. Here we go. Membranes. We're going to beat this to death. Membranes separate their cells and organelles from their surroundings, and they maintain a stable internal environment. Very important for homeostasis. Membranes are what we call selectively permeable. They're selective. They choose what goes through the membrane. Permeate means go through. Selective means you're choosing, right? So it allows some substances to cross more easily than others. And the way that, well, here it comes again, the way that the cell membrane is built allows it to be selectively permeable. What do I mean by that? Well, it's because this is a phospholipid bilayer. 2 layers of molecules called phospholipids. We may or may not have talked about this a little bit in Unit 1, but phospholipids are a type of lipid with a phosphate group. But the plasma membrane, we call it a fluid mosaic model because most of it is a phospholipid bilayer, but it's peppered and embedded with proteins in the bilayer. There might be enzymes, they might be receptors, they might be other communication molecules, they're channels, they're pumps. There's a whole bunch of different types of membrane proteins. But one of the main parts, in fact, the main part of the cell membrane is what we call the phospholipid bilayer. And the properties of the phospholipids are one of the things that helps it make it selectively permeable. Phospholipids constitute most of the membrane. This is what a phospholipid looks like. It's a phosphate group. It has a glycerol and 2 fatty acid tails. So it's almost like a triglyceride, except it only has 2 fatty acids and a glycerol and has a phosphate. But a phospholipid, what's special about a phospholipid is that it is amphipathic. Amphipathic. So an amphibian, right, is an organism that can live in water and on land. Amphipathic means it's polar on one side, non-polar on the other. Now, this is not a polar molecule. Remember, polar molecules are like I have a slight negative charge on one side and a positive, slight positive charge on the other side. That's what makes it polar, right? This is different. Amphipathic means there's a polar side and a non-polar side to each of these molecules. It has a hydrophilic, okay, it means likes water, polar head, and a hydrophobic tail. The hydrophobic tails face in, and the hydrophobic heads face out. So that means that, hang on, where's my page? There we go. That means that the inside of the plasma membrane, inside of the bilayer, is nonpolar, and the outsides are polar. I made this myself. Tell me it's good. But yeah, so that means right now that some molecules are going to have a harder time getting through than others. Say, for example, polar molecules have a hard time passing through that whole nonpolar fatty acid section in the middle, while nonpolar molecules can. Remember, polar and nonpolar, they don't mix. All right, you put water and oil in a glass together, and they're going to separate out because they don't mix together. The oil is nonpolar and the water is polar, right? So polar molecules are not going to be able to get through that bilayer very easily. So as I said, the polarity of the phospholipid bilayer contributes to this plasma membrane selective permeability. So we're going to talk about what can and what can't go through in 2.5. Small, non-polar molecules can pass through the bilayer, like oxygen and carbon dioxide. Those are non-polar molecules, and they're also very, very small. But large polar and charged molecules cannot pass through, like water, water in small amounts, glucose, ions, and other charged particles like that. Okay? But there is still a way, okay, because cells need glucose, they need ions, they need water. There's other ways for it to pass through the membrane, and that's where those proteins come in. Channel proteins and transport proteins can be used to transport other molecules across the membrane because, like I said, cells still need things like glucose and sodium. So here's a carrier protein, here's a channel protein. If we need stuff from the outside of the cell, one way for them to get through is through one of these proteins. It's a little channel, it's a hole, that can open and close to allow certain molecules in. Okay, one of which is called an aquaporin. It's a channel protein specifically for water molecules. Water can only pass through small amounts of the bilayer. It is a very small molecule, so sometimes they can weasel their way through the nonpolar part of the bilayer, but not always, so that's what they have aquaporins for. Plant cell walls, I told you this is coming back. Plant cell walls do not serve the same function as plasma membranes. This is something I have to get through to my regular bio students all the time. They're not doing the same job as the plasma membrane. A plasma membrane is made of a phospholipid bilayer and proteins. Plant cell walls are made of cellulose and pectin, a polysaccharide, and pectin is a protein and provide a structural barrier and support. And they allow a plant cell to build turgor pressure, which we're going to talk about later. On to 2.6. All right, talking more about diffusion here. All living things have to exchange matter with their environments, like we talked about before. Everything has to take in food, everything has to eliminate waste, and once again, cells rely upon diffusion for that. And what is diffusion? Well, diffusion is the tendency for particles to spread out evenly, to move from an area of high concentration to low concentration, from where there's a lot of it to where there's not very much. It doesn't matter what it is, it tends to diffuse. You spray Febreze in a classroom, it's concentrated in the bottle, and then once you spray it out, it's going to spread out, and that's what diffusion is. And cells rely on diffusion for this to happen. Okay, one of which, when we call it diffusion across a membrane, that's called passive transport. Passive means you don't have to apply a whole lot of energy. You can just kind of let it happen. So passive transport is a natural movement of substances across a membrane from a high to low concentration. So this only happens when there is what we call a concentration gradient. And a concentration gradient, and my definition is that it's a difference in concentration, the amount of something per unit of volume across a membrane. So take a look at this one. It's kind of grainy here, but if I have small uncharged molecules, non-polar molecules, what can happen is that they can pass through the bilayer, right? But they're not all going to pass through the bilayer. They're going to partially pass through the bilayer so that it's even concentration on both sides. That's what passive transport is. And cells have to rely on that. But they can manipulate the environment. They can manipulate what goes in and what comes out in order to control concentration gradients. Okay, because this doesn't seem very reliable, just kind of like hopefully absorbing stuff from your surroundings in order to survive. It's not super reliable all the time, so cells have other systems to do that, right? Just like you can't, put your head on a textbook and hope to absorb all of your AP Bio knowledge, right? Osmosis learning doesn't work that way. Okay, passive transport does not require energy from the cell, which is pretty nice, right? You don't need to spend any ATP on that. Selective permeability of the membrane and active transport allow a cell to control concentration gradients, is what I just said. I'm getting a little ahead of myself. But yeah, okay, the cell's ability to control the outside versus the inside and the concentration gradients between them is really important for it to maintain homeostasis and stay alive. Control what goes in and out and make sure it's taking in nutrients and eliminating waste. Okay, so the opposite of passive transport is active transport. All right, what if a cell needs some ions? What if it needs some glucose? What if it needs some particles that can't really pass through the bilayer? Or what if it really needs to get rid of some stuff and make it go against the concentration gradient? That's what we have active transport for. But active transport does have a cost. You have to use ATP to move a solute across a membrane. It usually, like I said, it goes against a concentration gradient, so it goes from low to high concentration. It's almost like pushing something uphill, right? If you're going from high to low elevation on your bike, you can just coast down the hill, right? You don't have to spend any energy. But if you want to go from low to high, you have to exert some energy to push yourself back up the hill. That's kind of a metaphor for how passive and active transport work. Large polar and charged molecules can be actively transported. So 2 reasons why it works. 1, you can use ATP to go against a concentration gradient, and 2, you can make things cross the membrane that can't really cross the membrane on their own. Okay? Whew. All right, 2 types of active transport, exocytosis and endocytosis. This is different, right? This is not going to use a pump. This is not going to use like a channel protein. You're going to form a vesicle with a membrane, and this is what the endomembrane system does. The ER, smooth ER, rough ER, and the Golgi apparatus. This is what they do. They form vesicles, and they do this thing called exocytosis and endocytosis, 2 forms of active transport. And they use a vesicle, which is a small membrane compartment, to import and export large molecules or quantities of molecules. So this is what exocytosis looks like, right? If there's something the cell wants to take in, it can infold its membrane because the membrane is very fluid, and form a pocket, a little, that's endocytosis. There we go. And form a little pocket for the cell to take in that particle and have a little pocket for it to get sent to the endomembrane system. Exocytosis is the opposite. All right, if let's just say my rough ER made some proteins here and it wants to ship it out, it's going to put it in a vesicle, and then that vesicle can fuse with the plasma membrane with the outside of it, and then send that out to another cell. Okay, 2 types of endocytosis are phagocytosis and pinocytosis. Phagocytosis is taking in a large molecule or even a cell, so a lot of your immune cells. If they find something they don't like in your body, they'll phagocytize them and basically take them in and dissolve them through phagocytosis. And pinocytosis is taking in large quantities of a fluid, okay, through endocytosis. All right. Moving on, here we go. 2.7 is all about facilitated diffusion, and that's passive transport aided by transport proteins, aka channel proteins or carrier proteins. So like I showed you before, ions in water can cross the membrane pretty easily through a channel protein or a carrier protein, and it'll diffuse just like with passive transport, except it can't go through the bilayer, so it needs a protein to let it through. So in other words, the protein is facilitating or making it easier for these charge or polar molecules to cross the membrane that normally can't. Okay, that's why it's called facilitated diffusion. They need a little help to get across the membrane, and that's what the channel proteins are there for. Okay, facilitated diffusion and active transport help a cell establish membrane potential. Now, might get a little complicated, but I'm going to try and keep it brief here. It's a voltage, or an AKA a difference in electrical charge across a membrane. Now, is a cell like a battery? In a way, yes. It can control one side of the cell being positive and one side of the cell being negative, very much like a battery, right? You hook up a battery to something, it connects the positive end and the negative end of the battery, and then it generates a current. It can do work, and a cell can actually do that as well. It can use a difference in charge on the outside and inside of the cell in order to do some jobs, in order to do some work. So membrane potential is established by positive and negative ions, right? So a cell might put in a whole bunch of sodium on one side and a whole bunch of potassium or even chloride ions on the other side, and make a difference. You can see like the outside of this membrane is positive, the inside is negative. It has a membrane potential. And a cell membrane potential that it likes to stay at most of the time is called resting potential, and that is a charge difference of negative 70 millivolts. Right, so if you pick up a battery, it's going to say, like a AA battery, it's going to say 1.5 V. That's 1.5 volts. That's the amount of charge difference between the positive side of the battery and the negative side of the battery. The charge difference between the inside of and outside of a cell is negative 70 millivolts. And milli is 1/1000th of a volt. So it's not very much, like it's still a battery, all right? There's still a charge difference, and it can use that to do some work. That's what we call an electrochemical gradient, a combination of electrical charge difference and concentration gradient difference. All right, so if you got a lot of ions on one side and not a lot of ions on the other side, there's 2 reasons why those ions want to go across and move to each other. 1, because of a positive and negative charge difference, right? Opposites attract. And 2, because of the concentration gradient, okay? Because they want to spread out from where it's more concentrated to where it's less concentrated. So diffusion and electromagnetic forces are both wanting to bring those molecules across the bilayer. In order to make membrane potential, we got to do some active transport, and that's where electrogenic pumps come in. Electrogenic pumps use ATP to actively transport ions in or out to generate a membrane potential or voltage. There's 2 of them that you need to know. One is called a sodium-potassium pump, aka sodium potassium ATPase, which uses ATP, like the picture has here, to move sodium ions out and potassium ions in to help establish a resting potential. Okay, so check it out. It's going to use ATP, active transport, to pump out sodium ions and pump in potassium ions in order to maintain what's called a resting potential, which is kind of counterintuitive. You're using energy to maintain resting potential. That's actually what your neurons do all the time, and that's why your neurons use a whole bunch of your ATP. So that is one electrogenic pump. The other type is called a proton pump, and proton pumps use ATP or electrons getting passed from one protein to the next to pump hydrogen ions across a membrane. Okay, and that can create what's called a proton gradient. So co-transport is a special type of active transport that uses electrochemical gradients to bring in other molecules like sucrose and glucose. Sucrose and glucose cannot pass through the biolayer. They're kind of big, they're polar, so they need what's called a co-transporter or a symporter here. So here's co-transport. All right, it looks a little complicated, right? But here's ATP being used to actively transport a sodium ion out, okay? And then this sodium ion can, its way to get back in is through what's called this symporter or co-transporter. And this symporter is like, okay, sodium, if you want to get back into the cell, you got to bring glucose with you. So it's like, I kind of use this metaphor with my class, like, all right, if you want to get back into the club, you got to bring a friend. So yeah, if the sodium wants to get back in, it has to bring in glucose, and that is a type of, that's a way for a cell to bring in glucose, which is super important, right? Okay, so that's co-transport. Now, 2.8, this is another doozy here, osmosis and tonicity, and I'm going to try and go through fast. Osmosis is the diffusion of water, okay? We talked about diffusion being the tendency of particles to spread out evenly from high to low concentration. We have a special word for it. It's called osmosis when it comes to water. Okay, so osmosis is super important because all living things need water, and they need to control how much water they have and how much water they lose to their environment. Right? You don't want to have too much water. You don't want to have too little water. You want to be just right, whether you're an animal cell or a plant cell. So osmoregulation is super important. It's the control of osmosis through solute concentrations. Okay, and osmosis follows a few rules here. Water will always diffuse from where there's less of it to where there's more of it. Okay, just like diffusion. Okay, but here's the thing. All right, water osmosis, the motion of that is also controlled by how much solute there is. Okay, what's a solute? A solute is a solid that is dissolved in water. Water is a solvent. Solutes like sugar and salt are solutes that get dissolved in water. Okay, sugar and salt, aka electrolytes, control where water diffuses as well, where osmosis occurs as well. So here's a little U-shape. This is a classic experiment here. If I have a semi-permeable membrane that only water can get through, okay, I have pure water on one side, and then I have water with a solute on the other side. Where is water going to diffuse? It's not evenly concentrated on each side. Water is going to follow where the solute goes, okay? And if you let this sit for long enough, water is going to go to the other side, okay? The solute can't get through. Solute's going to stay over here, so water is going to go join it. And yeah, that's what osmosis is all about. There's less concentration of water on this side than there is on this side, so water is simply, again, going to go from where there's more of it to where there's less of it. And where there's less of it tends to be where there's more solute. Okay? That's a huge important thing for osmosis. Now, the amount of solute in a cell or in a cell's environment determines whether it will gain or lose water. And it's super important to control that, right? Because all living things need water, and they need the right amount of water. So cells in a hypotonic solution will swell with water. That's not good. Well, unless you're a plant, then it's good. But hypotonic solutions will swell with water because there's more solute in the cell than in the surrounding solution. So if you're absorbing too much water, if you're an animal cell, you'll burst, right? But if you're a plant cell, that means you're absorbing more water than the environment, and that's usually good because you're building up what's called turgor pressure, which allows the plant to stay upright. Okay, so animal cells do not want to be hypotonic, sorry, and plant cells do want to be hypotonic. Okay, hypertonic is what nobody wants. Cells in hypertonic solutions will shrivel or shrink from water exiting the cell because there's more solute in the surrounding solution, right? If there's more solute outside the cell than inside the cell, water is going to go whoop, going to go follow the solute, okay, and then it will shrivel up and die. And that's not good for an animal cell or a plant cell. They're both going to shrivel up, or a plant cell will the word is plasmolyze if that happens. Okay, so if you're an animal cell, what you want to be is isotonic. Cells in isotonic solutions will have no net gain or loss of water because solute concentrations are the same on the inside and the outside of the cell. Okay, so there's no net gain of water. There's no net loss of water. Water's still going in and out. Okay, but there's not more of it being taken in or taken out. Right, so that means it's pretty balanced, and that's what you want to be at if you're an animal cell. You're not going to blow up. You're not going to shrivel up. That's what you want. Okay, now the second part of 2.8 is about water potential. Osmosis is determined not only by solute, I lied to you a little bit, it's not only determined by solute concentration, it's determined by pressure. And water potential is a property that predicts osmosis based on both solute and pressure. And here is the formula for how to calculate water potential. This is the Greek letter psi. Water potential equals solute potential plus pressure potential. Okay, and then solute potential is this is how you calculate solute potential, and this is what the main focus is in AP biology. All right, and the thing about water potential, when you calculate it, water moves from an area of high water potential to an area of low water potential. All right, and that is something to remember. And again, we're not going to be able to do practice problems here. Okay, if you want to check out some practice problems, I recommend going to my 2.8 part 2 video to work out some practice problems for water potential and get more details on it. But again, this is a recap, so we got to keep moving. This is the water potential formula. Water potential equals solute potential plus pressure potential. Pressure potential is at 0 when a solution is in an open container. Okay, so that means that the normal amount of atmospheric pressure is not going to exert any extra pressure. It's not going to affect osmosis any bit more than normal, right? So if you come across a practice problem where it's like a cell is in an open container, the solution in the open container has 0 pressure on it, 0 pressure potential. Okay, and then the other formula that you're going to have to know, but you're going to be given it, but you have to know what it means, and you're going to have to know how to apply it, is psi s equals negative ICRT. Psi s is solute potential, okay, the effect of which solute affects how water will go in and out. Negative is negative. I stands for the ionization constant, and the main thing that you have to know is that I is 1 when the solute involved is sugar, 2 when the solute involved is salt. Okay, salt dissolves into 2 ions, sugar dissolves into 1. That's where that comes from. C is the concentration of the solute in moles per liter. So that's the unit that you're going to use, molar, for concentration of solute. R is always the same number. It's I believe it's the what's called the pressure constant, but anyway, it's the same number every time. 0.0831. Okay, so that's what you're going to put in every time. Plug and chug. And then T is temperature in degrees Kelvin. All right, so if a practice problem gives you gives you a temperature in degrees Celsius, you have to add 273 to that to make it degrees Kelvin. All right, and again, we're not doing practice problems here. Go to my 2.8 part 2 video if you want to practice those. All right, 2.9, we're almost done. Okay, passive transport. This is kind of a review of our 3 different types of transport. First of which is passive transport. That is diffusion across a bilayer. It involves small nonpolar molecules, and it moves with a concentration gradient, or what I like to call downhill. Go from down, high to low. Okay, you don't need to spend energy to do that. So there's what I like to call downhill. Okay, and there's no energy required for passive transport. Facilitated diffusion is a type of passive transport, but it needs the help of a carrier or channel protein, which is what it makes it facilitated. So small charged or polar molecules, those that cannot normally pass through the bilayer, need facilitated diffusion. So like ion channels and aquaporins are involved in facilitated diffusion. Again, with a concentration gradient downhill, they just need the help of a carrier protein or a channel protein. No energy required for that. But active transport is the opposite. It does require ATP and membrane proteins. And it requires, it allows us to move large polar and charged molecules, and it moves against a concentration gradient from a low to high, or it has to pump it uphill, use some ATP to make it go uphill rather than just coasting downhill. Okay, electrogenic pumps, co-transporters, or symporters as they're called, endocytosis and exocytosis are all forms of active transport. Okay, last 2 topics, here we go. 2.10. Going back to the beginning, it's full circle, right? Prokaryotes do not have membrane-bound organelles, as we already said. Eukaryotes do have membrane-bound organelles. Okay, so the word that we use to describe eukaryotes is compartmentalized. They're compartmentalized, they're organized, because different functions occur in different parts of the cell, which are separated out by membranes. Eukaryotes have a lot of membranes. Prokaryotes don't. Okay, and that's what makes eukaryotes compartmentalize. Membranes separate out everything. Hey, this is your job over here. There's a membrane around you. Hey, this is your job over here. There's another membrane around you. Your job is to make ATP. Your job is to hydrolyze food particles and other particles. Right? You are separated out by membranes. That's what a eukaryote does. But prokaryotes, they're much smaller, and just everything kind of happens all in the cytoplasm. Compartmentalization is an advantage for eukaryotes because different reactions can occur without interference or competition. Everything is sectioned off, like I was just saying. And also, membranes allow for a greater surface area. Remember that? Remember that surface area to volume whole thing? Okay, the more membranes means more surface area. Surface area in eukaryotes allows for more reactions and better exchange of materials with the environment. Whether that's the intracellular or the extracellular. Intracellular means within the cell, or extracellular means between the inside and the outside of the cell. More surface area is better. Okay, we talked about that in 2.3. All right, we've been over that. So yeah, that's the two reasons why membranes in eukaryotes are a really good idea. Okay, everything's sectioned out, and you got more surface area for reactions. Okay, how did this come to be? Endosymbiosis, which is a little preview of evolution a little bit. Endosymbiosis theory explains how eukaryotes and compartmentalization evolved, and here's a nice little diagram of it. Here's a proto-eukaryote, a super old prokaryote, really. This happened approximately like 3.5 billion years ago. Infoldings of the plasma membrane allowed it to have endomembrane components. And then also the first endosymbiotic event is that an aerobic bacterium, one that can, I don't know, use oxygen to make ATP with glucose, was kind of swallowed up by another by this older cell. Okay, and then that evolved into mitochondrion, and then a photosynthetic bacterium kind of was swallowed up by this other cell too, one that can take light and convert that into, or store that energy from light into glucose. That was swallowed up by a larger cell, and they decided to live together, and it sounds crazy, but that's how the chloroplast evolved, right? So as I put up here, the early ancestor of eukaryotes engulfed another prokaryote with the ability to perform aerobic respiration, hence became the modern-day mitochondrion, right? So like, hey, oh, you're really good at making ATP. I'm going to swallow you up, and I'm not going to dissolve you. I'm not going to eat you because you're really good at ATP, so you can just live inside me. Again, crazy. You can just live inside me. I'll protect you. I'll maintain a stable internal environment, and you can make a bunch of ATP, and we'll both live happily ever after. And that's the evolution of the modern-day mitochondria. I know it sounds really dumb and crazy, but like this makes a whole lot of sense, and I'll show you why just in a second. Hey, same thing happened with the chloroplast. Hey, wow, look, you can use light to store that into a glucose molecule. That's awesome. Again, they live inside the other cell. Endosymbiosis. Endo means within. Symbiosis means living together. So they live together inside one another. And this photosynthetic bacterium, this cyanobacterium that was kind of swallowed up by the bigger cell and decided to live inside of it, that became the modern-day chloroplast over millions of years of evolution. Okay, and how we know this is actually true, the evidence behind this, is that mitochondria and chloroplast, they have their own DNA. It's like they're their own separate cell. They have their own ribosomes. It's like they could live on their own. Again, they have circular DNA, just like bacteria do. They have two membranes. We talked about that in topic 2.2, how they have an outer membrane and an inner membrane. It's like, hey, that outer membrane is probably the one that they, we just talked about phagocytosis, taking in a larger particle with a vesicle. Okay, that outer membrane is probably a remnant of that vesicle that they were taken up by. So the two membranes make sense again. And last piece of evidence that they were once their own, or mitochondria and chloroplast were once their own separate organisms, is that they can reproduce on their own. So the whole steps of mitosis that a cell follows, mitochondria and chloroplast don't really care about that because they separate and they divide on their own without any help from the rest of the cell. Hey, whew, that was Unit 2. Wow. All right, let me know if you have any questions, or you can check out the more specific topic videos. Either one works, but just comments if you have any questions, and we'll see you next time. We're going to do Unit 3 here pretty soon. So, bye.